remarks, I'd now like to hand over to Dyann Wirth, who will give her perspective on the situation.
Well, thank you, Alan, for the introduction, and I really want to thank DNDi for holding this event to celebrate what I think is a really important milestone in its history. And I'm happy to see many of the friends from the very early days. Bernard, thank you very much for bringing together all of this group, many of whom I haven't seen in the last 5 or 6 years. So it's a real delight to be here. And so let's see, we've Did I do something? Let them put it up, okay. So I've been asked to talk a little bit on the challenges that we still face, and what I've decided to do is look both at the situation that existed when we began the discussions of the need for an organization like DNDi, and then the changes that have occurred in the field since then. Well, we'll see what happens. I could talk without slides. Alan said, I did have slides. But so in the early 2000s, there was really a situation in front of us, here we are, where in fact, this is sort of a kind of where we were with infectious diseases. Many of you remember this was the SARS epidemic. This was an op-ed piece from the New York, presented in the New York Times, sort of putting SARS in perspective with the many neglected diseases that appear here. But if you remember, there was this worldwide outcry, we need to cure SARS, it's going to be an epidemic, a pandemic, and I think this emphasizes that, in fact, once diseases become part of the psyche of the world, enormous effort can be put in there, it can be put forward. And so I think that's really the concept of DNDi. It's both the raising of the awareness and then bringing the best science to bear. And if we think about where we were in 2003 when DNDi formed, the drugs for the diseases that we're talking about were old. Most of them were discovered in the first half of the 20th century. As many of you know, they have a limited therapeutic window. Many of them are almost as toxic as the disease itself. And of course, for most of them, we did not know the mechanism. And I think more importantly, these diseases had completely missed the revolution in drug discovery. Science discovery, that is, the science in these diseases, was actually some of the most advanced in eukaryotic protozoa, but drug discovery was completely uncoupled, or almost completely uncoupled, from scientific discovery. The technology lagged, and there was a very fragmented effort. And I think this is just illustrated. I'm going to talk a little bit about genomics today as sort of one of the combining themes which I think has changed the field. So this is actually the issue of Nature commemorating the 50 years since the discovery of the double helix. And the top line is sort of through 1950 through about 1990, and I place on this panel the drugs that existed for some of the diseases we've been talking about and when they were discovered. In fact, at that time in 2003, atovaquone was the newest of the anti-malarial drugs, and it had been discovered in the 1950s. So there was really this disconnect between all of the development in science and drugs for the diseases that we were interested in. But things changed, and in fact, there's really been a leapfrogging into the 21st century during this last decade. And so, and I attribute that, and this is, my opinion, is to the genomic revolution. Essentially, you could genetically engineer parasites, you could do whole genome sequencing, to the sort of the idea that new chemical entities have been identified. There's been success in developing assays There's been success in high-throughput screening, and we heard about that yesterday. And there's been this exploration of chemical space in these diseases. This is something that's happened in the last decade. And we've been combining genomics and chemistry, so there are new ways to identify targets, and there are tools, and I'm going to demonstrate this with RNA interference, but this is just one of the tools that allows us to begin to dissect the biology of these organisms to understand where their Achilles heels or where their choke points are. And I think that's going to be critical going forward. And I would also say that, and it's been emphasized by the previous speakers, that there's now a concerted scientific effort on the problem of drug and vaccine discovery in the area of neglected diseases, broadly writ. And I think DNDi is one of the core organizations that has led that that idea that scientists need to work on this. It's not enough just to solve a fundamental and interesting scientific problem, but there is a need in these diseases to translate, and that, I think, is driving a lot of the work. So I'm now going to give you a few examples of what I think are kind of breakthrough technologies that have changed the game in the broad field of infectious diseases. And so here's just back onto this panel. Genetically engineered parasites. So while there were no drugs being discovered, parasites were genetically engineered in the early 1990s. High-throughput screening in these groups began being developed in the early 2000s. RNA was discovered in T. brucei in the early 2000s. And the first genome sequences of all of the organisms we're talking about were published in the early 2000s. So the tools were in place, and now the idea was to apply these. So probably one of the most, in some ways you think about this as breakthrough technology, but it's not. It's a technology we've had around for a long time. That is the measurement of killing parasites with a compound, whether those are parasites in a cell, such as the malaria parasite, where today over 5 million compounds have been tested in this live dead assay. Does a compound kill the parasite? And it's been an unbelievably fruitful way of identifying new chemical entities that kill the parasite, and subsequently the identification of new targets of those chemical entities. And we're beginning to see this. This is clearly happening in the diseases that are a focus of DNDi, but I would argue that this particular assay, that is the development of high throughput, very broad-ranging assays that look at all of chemical space, is extremely important and something I know is going on, but something that should really be accelerated. And there are great assays. Genetically engineered parasites now allow you to use imaging as the way to read this out. You can label parasites with fluorescent proteins. This is a parasite that only expresses the fluorescent protein when it's in the intracellular amastigote form and not at any other stage. So you can identify drugs that kill only the form that you're interested in. So you can develop very specific assays. And this technology breakthrough, I think, is extremely important, and one that we're beginning to see fruits be born in terms of identification of new chemical entities with specific targets. The other technology which has particularly changed the trypanosome field is the ability to, in a very precise way, modify the parasite's genome and either independently knock out or remove genes through RNAi is probably the technically most useful way of doing it. And that allows you to study the role of a gene very directly and decide whether it's a target, or in fact, in sort of more broad-ranging technology, actually make a library in which you attempt to inhibit all of the genes one by one. So each single organism has a different gene inhibited, and then ask, how does that change the response to a drug or the response to a nutrient? And in this way, you can begin to identify what I think is the fundamental thing that's still missing from much of what we do in this field, is the fundamental choke points or Achilles heels of the parasites. There have to be many in Plasmodium now. There are probably a dozen new targets in Plasmodium falciparum, and I expect there'll be the same number, perhaps more, in the kinetoplastid parasites, and this is something that I think really needs development, and the tools are there. Just one example from our chair's laboratory, in which he's identified pyridoxal kinase as a key enzyme. He can eliminate the gene in a sort of specific way, and all the parasites stop growing in vitro. And interestingly, they also, the parasites are also killed, killed in the mouse. And so I think it's a very interesting and important time where we can actually begin to catalog everything that's important to these parasites. Okay, I think the other key, if you're going to use the strategy of phenotypic screening, that is doing whole cell screening and asking what kills the parasite, is you need a way to identify the target, because as you heard yesterday, for those of you who were here, as you begin to look very specifically at drug development, having the target is extremely important. And if we're actually going to use all of the knowledge and all of the advances in chemistry, in biology, in pharmacology, then knowing the target greatly enables our ability to do that. And again, I would say here, genomics has provided so far the best tool we have in the toolbox. And I think that's through the selection of resistant parasites. I'm going to show you an example from Plasmodium, which is from my own work, or from the group at Harvard, but I think this kind of example can be applied to many of the parasite systems that we're working with today. And part of the reason that one is able to do this is shown on this diagram. This is the sequencing explosion. And if you look at this, at the time the human genome was sequenced, it cost about $3 billion. At around the same time, the first parasite genomes were sequenced, and they cost in the $30 million range. Today, as we speak, it costs about $300 to sequence an entire Plasmodium genome, and in fact, using kind of multiplexing and so forth, that number probably drops into the double digits quite soon. And so that changes the paradigm of the kinds of experiments you can do. Now that my graduate students don't ever, if they think they might have confused strains, instead of doing any experiment, they just send them for sequencing, making me go, what? We have to sequence this to know whether you made the right construct. But I think it's very important to understand that this changes the paradigm. It also changes the paradigm of what we can look at in the field. So, we had a discussion yesterday, there were several issues raised about whether the parasites, the organisms that are being used in lab experiments, relate to the parasites that actually exist in the field. And sequencing actually gives you the tool to look at that with great specificity and detail. And so this is, I think, a sort of game changer in terms of how we approach things. So I'm just going to show you an example of how we do target ID. This is target ID of a molecule that's been known as an antimalarial for a long time. It's derived from the plant derivative febrifugine. It's helifuginone. It's a very potent antimalarial, but the target was unknown, and it had significant side effects. And so what we do is simply subject parasites in culture to stepwise resistance selection. And amazingly, this occurs quite easily. This has now been applied, I would say, for dozens, maybe in the low double digits of compounds, and in almost all cases, we've been able to isolate mutants, we and many others in the field. So this is a very straightforward experimental technology. And then, so this is what we call select-seq. So you take the wild-type parasite strain, you put it under drug pressure, isolate resistant parasites, and then you simply sequence the two genomes and compare the sequences. And you can see this is just an example. We do independent selection so that we have independent confirmation. And then you sequence. And in this case, in essentially a single step, we identified the target of helifuginone. It's the prolyl tRNA synthase, an important molecule in protein synthesis, and a choke point in the parasite's amino acid utilization and starvation pathways. And so again, this now gives us the opportunity to explore this as a specific target. And I would posit this is likely to be a target in any of the parasites we're talking about today. And so, sorry, I went backwards. So I think that gives you a sense that really this is leapfrogging the technology. So instead of taking years to identify targets, you can identify targets in weeks to months of finding a compound that kills the parasite. Now, of course, and Tim Wells will know this, one of the issues, of course, is if you can isolate resistance in vitro, then what does that mean for the field? And I think, and the utilization of drugs. And I think it's very important to realize that what we can do in vitro doesn't really reflect what happens in vivo. And I won't have time to talk about this today, but as we think about new drug development and new ways of approaching combination therapy, I think we also need to think about the possibility that resistance will emerge. I would posit that, in fact, resistance hasn't emerged in large part for the kinetoplastid drugs is because they're really not very good drugs, and they're not putting very strong pressure on the parasite. In contrast, the antimalarial drugs have been very strong selectors, and in each case, the resistance to antimalarial drugs, including the recent reported changes in sensitivity to the artemisinin compounds, I think tells us that resistance is something we have to think about. And we should start thinking about resistance as we are thinking about screening and developing new drugs. And I think it's very important to keep in mind that this is a biological property. Someone said earlier today, there's this evolution of the parasite in the host, now, and obviously an evolution of the host in response to the parasite. So resistance is just part of evolution, and we should recognize that as part of what's important in terms of thinking about our new drug discovery. And I think it's important to keep in mind that resistance and fitness of the organism in the absence of drug are extremely important balancing or evolutionary or selective forces. And one of the concepts which we're exploring in Plasmodium, but I think is equally true in any other infectious agent, is the idea that in fact a resistant organism creates a new target. And it may be possible, and we certainly demonstrated this in laboratory experiments in Plasmodium, that if you have a resistant parasite to one drug, it can make it sensitive to a second drug. And if you're lucky, there's actually an evolutionary trap that occurs where resistance to one drug creates sensitivity to another, resistance to that second drug creates sensitivity to the first drug, as if you put the organism in an evolutionary loop. And I think this is an important thing to think about going forward, because it is true that if you expose billions of organisms to a strongly selective agent, resistance, if it can occur, will, and we need to be planning for that. I think the other important thing for the challenges for the next decade, I'll be a little controversial here. I'll say that we have the technological breakthroughs necessary to develop new good drugs for acute infections. Clearly access, clearly development, clearly animal models, many of the things that we've heard about are critical to this. But I think conceptually and proof of principle, compounds exist in the world that will kill these parasites, and they will be translatable into drugs for the acute infection. I think the next challenge is the need for solutions to latent chronic infections. So Chagas is one that this group deals with, Plasmodium vivax, hypnozoite stage, others, TB, perhaps leishmaniasis, latent or chronic. And I think that this is going to cause us to go to a different kind of approach, and that is the host-parasite relationship. Obviously, immunology plays an important role in the immune response, but I think also metabolism and nutrition. And we heard a little bit about this yesterday in the scientific symposium, and I just want to sort of posit one kind of example here as a last example of kind of a way of thinking about how one might approach this. And so I'm going to use work from Barbara Burley's group in my department. This is to look at chronic T. cruzi infection. As you all know, it's the intracellular form, the parasite's in its intracellular form. It's the form of the disease for which drug development is acutely needed. And actually, we know relatively little about the biology. And so Barbara's group has taken the strategy of saying, in fact, a parasite living inside a host actually plugs into the host metabolically in quite a significant way. And by using this genome-wide RNA screen, now focusing on the host cell, she's been able to actually identify pathways that are critical for the parasite to maintain its infection in a host cell. And in fact, this is just a part of that work, but in all of the pathways in yellow, if those pathways are knocked out, you see a decrease in parasites in the host cell. And in the blue, actually, if you knock out those genes, you actually see a dramatic increase of the parasite in the host cell. So clearly, the host cell genes are controlling the parasite as it grows. And so, in fact, this is a whole new way of thinking about chronic infection, obviously not one limited to our thinking, but I think it's one that the field needs to embrace. This now involves perhaps interfering with that interaction between the parasite and the host from the host side. There are certain advantages. Resistance is probably going to be less of a problem. But of course, the flip side is you're now interfering with a host cellular function. This will present its own challenges. But I think this is something we need to begin to think about as we go forward. And Barbara's gone on for the AKT pathway to do experiments to demonstrate that the initial screen, if you knock out AKT, you knock down intracellular parasites, and you can mimic that with a drug that targets the AKT pathway. And so, again, I think this is another way to begin to think about this, to begin to address perhaps the more challenging biological questions, that of latency, where the parasite is not growing or growing very slowly. I think finally I just want to make a couple of comments on what many speakers have said, and I think there's a change overall in the community, and that is that it's going to be very important to study these diseases in their natural settings, not just to implement what we discover, but actually to begin to understand the disease in its natural setting. One of the comments made yesterday afternoon was that, in fact, for many of the diseases that are the focus of DNDi, the epidemiological understanding of which strains are important in disease is really not there. And I think this is an area where one needs to do this in disease endemic settings. And it's important, I think, also that this is a way of translating the knowledge, to begin to translate fundamental knowledge of the natural infection from the disease endemic countries back to the laboratory. And I think this is a place where both training and kind of mutual learning, as Marcel Tanner has taught me, it's really mutual learning between scientists and clinicians working in disease endemic settings with scientists working in the more technologically developed settings that will have our greatest advances, because then we will be addressing those important questions. And I also think that technology is really creating the opportunity for innovation. If you think about it, this field of drug development for neglected diseases had leapfrogged into the 21st century. It's kind of like we didn't get a landline, we actually just went directly to cell phones and in fact to smartphones. And so I think it's very important that we kind of use our imagination to think about where we can go with our knowledge. And with that, I'll stop. The cures for neglected diseases, imagine the possibilities. I think this is an opportunity for the field to really think about what can we do. We know we can do it. We know that there are molecules out there that will meet our needs. We just have to find them and develop them. And it's a big challenge, but it's not a bigger challenge than we started with 10 years ago. And with that, I thank you.
Would you like to take a couple of questions?
Sure, I'm happy to take questions.
One thing, I mean, you talked about the power of genomics there in relation to drug discovery. You didn't mention the potential for diagnostics, A, for identifying infected individuals, and B, for measuring test of cure. Would you like to say a couple of words on that?
Exactly. I could have gone on forever about genomics, but I decided I should keep the thread. But I think diagnostics is extremely important, and I actually think that sort of smart diagnostic, that is, being able to detect resistance if that's an issue in the real setting, being able to detect organisms at very low levels using essentially molecule level detection is within our grasp. It's not there yet, but it's certainly something that we should be working on. So yes, enormous opportunities.
Okay, thank you.
Are there?
I think we'd better move on.
Okay.
I'd better move on in the interests of keeping to time and also getting opportunity for the members of the audience to ask questions after we've been through each of our panelists.